Those of you who haven't met, my name's Tom Wood. I'm from eastern Idaho, live just outside the town of Rexburg now, grew up in the metropolis of Newdale, just east of their population 300. Grew up on a potato farm, small grains, alfalfa. Dad quit growing spuds in the late 90s while it was still his decision to quit doing it. I ventured out into the custom farming realm after that, started a custom haying business first. Well, about 2003, my brother and I got into the chemical application things, started building potato row markers, doing mark out for guys in the spring. My dad had been a crop duster since about 85, somewhere in there. My brother has since taken over that business, still does that. Slowly over time, we built this application business up, still farming on the side. All the times we go, I still farm there. Somewhere along the line, about 2012, I stumbled into a seminar sort of like this. It was all new stuff, alternative. Nobody was talking about this trace mineral stuff and biology, and something kind of clicked in my head. So anyway, we went back and started playing with some of this stuff on the farm and saw some results. It was fun. It was something different, and input costs were going down and things were changing in the dirt. And in 2014, we had a weather event in my neighborhood, kind of all of southern Idaho, that we got into grain harvest, and it started to rain the 1st of August and really didn't quit for a couple of months there, made for a really interesting harvest. And we had a situation where we have a condition in cereal grains that you probably heard, vomitoxin. It's caused by a couple of organisms, Fusarium graminearum or Fusarium culmorum. Those organisms produce a metabolite. There's a stuff that they excrete called deoxynivalenol. That is the organism, or that is the compound that is vomitoxin. They call it that because when you get the concentration levels high enough, if you feed that grain to hogs, it will actually make them vomit. Vomitoxin. So that's where it got its nickname from. The Dawn is the nickname for it, vomitoxin Dawn. The Dawn levels were sky high that year in the cereal grains. It was just off the charts. And it was interesting, my brother and I noticed as we made our fumigation run, we would start kind of south from where we were at. So I would usually do the Telone, he would do the Metam products, and we would start further south and work our way up home, up to make the loop. That most of the guys, pretty well, it was a general statement that anybody who had been a heavy fumigator had a way worse vomitoxin problem than the guys who didn't fumigate. And it was interesting, the more that you got dealing with guys, talking to neighbors that were more into the biological side of things and aware of their biology, whatnot, that vomitoxin situation decreased as microbial activities and farming practices increased. On our own farm, not trying to pat but we came through scot-free. I had absolutely no problems with the Dawn whatsoever. Falling numbers were good. We dodged a bullet that year. I just got curious that winter about this Dawn, how it had blown up out of nowhere. Like you'd hear about it occasionally here and there, but nothing broad scale across the board like it did that year. So somewhere along that winter, I came across a research article talking about a compound called Chitosan. Never heard of it before. This article was talking about how effective Chitosan is against this Fusarium graminearum and Fusarium culmorum. And at that point, the hook was set, and we fell down the rabbit hole at that point, and I've continued on. I spent the next year and a half researching this. I've been through volumes of research papers. I got to where I could talk this compound pretty fluently. I knew it very intimately, and I was trying to find a product now. I knew the compound, I knew what this was, I knew what it was capable of, but I needed a product to play with. So again, the miracle of the internet in this day and age, I spent hours, made numerous phone calls. There's companies out there that would have a vague description without naming what it was, and I knew what they were playing with by what the claims they were making. Talk to them, and they would talk about the effects that this was a colloidal chitosan. But I knew that was still a cluster. Like we were always looking at Dalton size and molecular weights trying to get this in. I didn't want that. I talked to guys that had chitin. We'll get into this in a minute, but chitosan is a derivative of chitin, but they are nowhere near anywhere the same thing. Just divine chain of events, maybe, I guess. I was on the internet one night in a farmer chat room. I spend more time on a couple of those than I'll admit to anybody. And there was a gentleman on there that had mentioned the word chitosan. The thread topic was, what are you doing to increase wheat yields? And this guy had replied back that he's seen a huge response from a big micronutrient package and a chitosan product. I sent him a message. There was another human that knew this word, like there was a kindred soul out here. So I reached out to him. We went back and forth a time or two. This gentleman actually passed on my information to Mark and Robin, and they just cold called me at home one night about 8 o'clock. We talked till midnight. Sent me research articles the rest of the night at 6 in the morning. I still hadn't been to bed. Called them back, told them this is what I was looking for. So we bought a pallet. We started playing with this. Spring of, can't remember where we're to now, 15 or 16 was our first time playing with this. We didn't know what to do with it. These guys, we didn't have a lot of exposure in the West at that point. Hadn't been into a lot of the Western crops. The company's based out of the South. Our manufacturing facility is just outside Lafayette, Louisiana. Mr. Nichols resides in northern Georgia. Our president is in Mississippi. One of the other sales guys is in southern Alabama. And the science team is, they say, just outside Lafayette. So I was kind of lone dog, so we didn't know what to do. So we just brought in the pallet. We mixed up a pint per acre in our sprayer. We've got, like I say, we can apply anything under the sun at our place. But so we went out and just sprayed a pint per acre, broadcast, pre-plant, before we even spread fertilizer, did anything. That was the first thing we did, and away we went. And then didn't see a damn thing all summer. There was really nothing significant in tiller counts, kernel counts once in a while. I thought I'd see something different, but we had basically about given up on this, that this, just didn't do anything. Thank goodness for yield monitors, or we'd have missed it, because we'd kind of quit looking for stuff by the time harvest rolled around. We were growing all malt barley for Anheuser-Busch on the farm that year, and when we went back and looking at the data, right to the last sprayer nozzle, the first field that we had done this on, we had a 7 bushel to the acre increase. The next field was a 19 bushel to the acre increase, an 18 bushel to the acre increase, and the field number 4 was a 21 bushel to the acre increase. So we blown away at this point. We got noticing for the next couple days too, sunrise, sunset, you could actually see right to the sprayer nozzle where we'd sprayed this out earlier, because the stubble would have a different color to it, kind of a pinkish purplish hue to it. Though we think it was probably due to some micronutrient translocation into the plant that caused that. We noticed you could go out with a pair of scissors, make a clean cut out of the test plot and out of the check plot, and hold them up, and you visually could see a thicker, heavier straw in this. So at that point, we were hooked. We've been using this, playing with this ever since, and that's how I got here today. These guys approached me a couple of years ago, asked me if I would consider coming to work for them, and I told them no the first time, and they asked again and said please that time, and I said okay. So I stepped away from the application business. My brother's still running it, so we're still involved with all this. Like I say, we mark out spouts of fumigation. We got a 502 air tractor at the place, 1184 rogator. Anyway, we're still involved here. So chitosan, what is it? Where does it come from? What does it do? And why should you use it? So this is going to get a little bit technical for just a minute, but this will start to shed some light into the differences between us and what sets us apart. Chitin is one of nature's building blocks. Anything that you find in nature that has an exoskeleton will have a chitin component to that. That will be made of chitin. So shrimp shells, lobster shells, insect shells, the mouth parts on nematodes, the stylet on aphids. This is all chitin construction. Chitosan is a derivative. We start with the chitin component, and then it has to go through a process they call deacetylation. Basically, we take this molecule in, and there's an N-acetyl group attached to this molecule. We cleave that off, get rid of that, and we're now left with chitosan. This is where some of the other competitive products will stop. we continue on with the process here, that we now start to tear this down for molecular weights, because science knows, this compound's well known in the high, the big circles in agronomy here. And it's been proven, they know full well what this does, and they're figuring this out now to figure out what, they know that a certain molecular weight will have a certain activity on a certain organism. So you start on the lighter end of the spectrum is going to be better for your fungicidal effects, bactericidal, virucidal, pathogen, things like that. As we gradiate up the scale, we get over to the heavy end of the molecular weights. These now, you pick up the efficacy on the insects, the larger organisms. We're going to get into more of the nematodes, soil-borne pests that way. There's other companies working on this technology that are trying to extract specific molecular weights because they don't want it to do everything. They know if they can take a specific weight, it will go after a specific fungal infection. Another specific weight will go after a specific bacterial infection. It gets extremely expensive when you start trying to extract for specific weights like this. And it's really easy to miss the target, because if you're after this exact thing and you miss the weights just a little bit, you completely missed your target. What we do is we tear this down, clear across the spectrum, and when we're done, everything gets put back in. So our products are a 2% Chitosan solution. There isn't another product on the market that's been able to obtain our level of concentration. And we span the gamut of molecular weights. We go from the lightest to the heaviest. So you're going to get the most activity possible out of our products. We are the most bioavailable form of Chitosan on the market at this point. So, and we don't anticipate any competition in that realm, well, really ever. What does it do? First and foremost, one of the big effects you see out of this is the SAR response, systemic acquired resistance or systemic induced resistance. Basically, we're going to turn on the plant defense mechanism. We'll get into a little bit of this, how this works. It's been shown, this is well documented. Don't take my word for anything that I say today. I might be completely full of crap. I want you all to go home and take this word and start Googling whatever's on your mind. Chitosan and insects, Chitosan fungal, Chitosan white mold, but whatever. Just Chitosan in agriculture. Go double check everything I tell you today. Been shown to be a very effective fungicide, bacterial-cide, viral-cide, insecticide, nematicide, plant elicitor. We will turn things on in the plant. This is where the nutritional side of things is critical though, especially the trace elements. We overlook the traces far too often, and they are so key in driving plant function. That you've got the NPK in the tank, but your micronutrients are the key in the ignition system. And without that key and that spark to start things, nothing happens. So we'll put this on, we're going to ramp up the plant, and if we don't have that in place, we're going to tip over. You'll see results, we'll do fantastic for a week, and then you're going to call me and I'm a dirty SOB because your snake oil didn't work. At that point, I'm going to tell you I want to see a plant sap analysis so we see what's in there, because anytime this happens, it's pretty universally there is a micronutrient deficiency going on somewhere. Go look into this. A big example of this would be molybdenum is catching some traction nowadays. If you don't always need more nitrogen, you've got to have molybdenum. If you don't have molybdenum to put that key in the nitrogen ignition, you can keep dumping nitrogen on and wasting your money, and you're not going to get it into the plant. You've got to have zinc, phosphate. This is another one that's catching a lot of traction these days. So we will turn things on, but we've got to have, we're not a nutritional replacement product, but we will drive function in the plant. A nutrient and mineral, microbial stimulant. This is a big one that we're going to, so the biology that you're going to put out here with your stubble digesters and whatnot, the mycorrhizal population that we all are starting to understand now how important this is. We're going to help with all this. We will become a food source for the good guys, and it will kill the bad guys. And I'll explain, yeah, I know, everybody's bullshit flag now is going to the top of the pole, but bear with me. We'll explain this a little bit here, how this is possible. Nutrient mineral chelator, increase the model conductance for transpiration control, will increase the photosynthetic rate in your plants, increase cold tolerance. Part of this, I think, Mark, everybody got a slide here from a potato situation we had in Idaho. Well, I got some more, we'll go over this in a minute, but fantastic things for cold tolerance, helping with the frost. Wetting sticking agent, fantastic. We started off as adjuvants because, and we've grown, been successful because it works very well that way. And as a food preservative. So how does it work? You guys want to read, we'll just quickly go through this. I'm going to kind of combine the fungicide, bacterial side, viral side, especially the fungus and the bacteria. As a fungicide, nature and the good Lord and divine intelligence, something that went on here, this is already pre-written into the system. Most of your pathogenic fungi are going to have a chitin component in their cell structure. There actually is certain fungal organisms that people will cultivate for chitin chitosan extraction, but they're actually trying to farm this stuff now out of certain fungal species. There is a direct effect that happens due to that chitin component within the cell structure. When the chitosan comes in contact with it, there is an attraction that happens, that it encapsulates the organism and they start to pull towards each other. When that happens, eventually you will start to tear that cell wall and it's going to die from cell lysis. We're just going to tear that apart and it's going to bleed to death. Sort of the same effect happens with bacteria. We're a little bit slower with bacteria because for the most part, bacteria will lack that chitin component within their structure. But at the point of cell division, the chitosan will actually, it's more of an electrostatic mode of action. With the bacteria, we will interfere at that point of cell division so that one side of the two organisms now will have an incomplete cell structure, wide open. The other side, generally there's some morphology that goes on that just makes that organism now inconsistent with life. So. Some of the beneficials in the fungicides, your aspergillus, mucor, mortarella, trichoderma. There's good guys. Some of these names you'll hear back and forth that you think that's a bad guy or you thought that bad guy was actually a good guy. Once you get into things, you'll have good guys and bad guys in basically every family as this goes through. Trichoderma harzianum is one of my favorites. This is one of the most prolific chitinase producers that science has been able to identify yet. And we'll get into the chitinase side of things here in just a few minutes. But bacterial side, again, the SAR response. Anytime that we put this out, we're going to trigger this self-defense reaction within the plant. Through this, we'll chelate some of the nutrients, prevent the organism from being able to access them. It will actually interfere with some of the biofilms, the metabolites that are produced by some of these pathogens. We'll actually bind up with those and render them useless. Some of your beneficials out of the bacterial world, Flavobacterium, Bacillus. Most of your Bacillus, when you go through this, they're really is some of your best ones you can put in the soil. Really powerful, this Cytophaga. Pseudomonas fluorescens is another one of my favorites. Clostridium, Streptomyces, Serratia. So again, the Clostridium, Streptomyces, you think that those are usually bad, but there's actually good in those too. And if you can increase the good, there's no room for the bad. Kevin was talking earlier here with you're trying to break down your residue, whatnot. That's the other benefit to this, is as you're putting the good guys out here, putting them to work, they're taking up this space. You have an environment in your soil. And nature never allows a void. She will put something in there, whether you like it or not. So if you're putting the good guys in and trying to take care of them and cultivate them, there's no room for the bad guys. So your plant health and disease, it's going to kind of automatically start to do this as you get some of these things right, get some biology back in your soil, get your nutrition in place where it needs to be, and there's kind of some magic that starts to happen. On the viral side of things, again, the SAR response, expression of resistance genes, jasmonate synthesis, stimulation enhances cell wall lignification, phytoalexin synthesis. This works more to inoculate the plant against a viral infection than it does to kill or stop the viral infection. But you guys, with the potato stuff, where I farm, we're right on the verge of seed production and commercial production. You go 5 miles east of me, it's all seed. From there west, the rest of the state is all commercial, so we kind of dabble, kind of play in both sides of the world. We've got a seed producer up the road that they got brave and decided they would try some of this a couple years ago. They took one pivot, split it in half, and we went on in, second week of July with an aphid spray. We put 1 pint of O2YS in on half of the pivot. The other side, same formulation, like I say, this is an insecticide application for the aphids. When they got those reports back from Idaho Crop Improvement, Idaho Crop Improvement will pull samples, send them to Hawaii for the winter grow outs. When they got those reports back the next spring, the PVY number on the side that we had treated, the one guy at the farm told me that the number had stayed the same from the previous generation. We did not gain at all. The farm manager told me that we actually reduced the PVY number just a little bit. It actually went down a little bit, which you guys are probably all well aware of doesn't happen. You just try to control this and keep the burn as slow as possible, but PVY is always going to escalate through the generations. The other side of the pivot, where we had the check there with just the same insecticide, the PVY number had escalated so far in that generation, Crop Improvement would not allow those tagged for seed. They had to go to the warehouse. So word got about this. We've spread now some of the seed guys in southern Montana. We're seeing the same results out of there, that we're holding or actually reducing the PVY number one generation to the next. Along with this now, the reduction in aphid sprays. When we get this population, when we get these levels up, there's a repellency effect that will happen first with the chitosan. If they do show up and decide to try to take a bite, why, it gives them a really bad day. This is how. Nematicide, insecticide. As the company has matured, we started off, like say, with in the adjuvant realm of things, that we would just make things better, kind of from market input, as what the customers and the grower base, the farmers across the country have told us they needed. We have continued to pursue registrations now. So we have our Nematicide product, you can see on the flyer poster over there. That is an EPA registered nematicide. We have full-blown EPA registration on that, and they don't just give those away. Your stuff has to work before you get one of those. With this again, so we'll get into this chitinolytic organisms and chitinase production. In nature, like I say, everything with an exoskeleton is made of chitin. So to keep the ocean from being full of dead shrimp shells and crab shells by now, nature has there's an enzyme for everything we talked about earlier here. For chitin chitosan, the enzyme is called chitinase. And there's a subclass of organisms that are termed chitinolytic or chitosanolytic. I stick with chitin because it saves me a syllable. And I say this a lot, so it adds up over time. But all that means, in order to meet this chitinolytic classification, is that they produce this enzyme chitinase. So the good guys that I was telling you here earlier, those are ones that we know have all been shown to produce this enzyme chitinase. Once this enzyme comes in contact with the chitin chitosan, it starts to tear it apart. You'll have endochitinases, ectochitinases. They will chew it in the middle, chew it from the ends. They start to break this compound down and tear it apart. Interestingly enough, your plants will also produce chitinase on their own. This is one of the compounds your plants will produce as a phytoalexic response. Your plant is capable of doing this. Once we get this chitinase level up, insects, it's again the divine intelligence that's put into all of this is just incomprehensible. But insects recognize this. They know anything that's been through a molting process and had to escape that chrysalis, that insect actually had to produce a small amount of chitinase in order to soften that chrysalis to be able to get out. Nature, they're all well aware of what this is. So number one, we've increased this SAR response. Your plant's firing, it's on a little different frequency now. As long as you, again, if you've got the nutrition in place, we talked about earlier the guys with the bugs see differently than we do. They recognize insects have a job in nature. They're there to take out the weak and the weak and the sick before they have a chance to reproduce and send that down in the progeny. They have a job. Nature put them here. They have a job to do. But when they recognize this, when we get the chitinase levels build up in the plants, A, the insects don't want to be there. B, if they do and they come in contact with this chitinase enzyme, it just starts to tear them apart. So again, the mouth parts on a nematode, chitin. Stylet on an aphid, chitin construction. Anything like this, the external parts of this comes in contact with the chitinase, it will just start to tear this down. We have found, it took me a little bit to figure this out in Idaho, nematicidal properties on this. We've learned that so soybean cyst nematodes, do you guys deal with those, contend with those at all in this neighborhood? Okay. This is one of my favorite ones to go after because of the life cycle and reproductive cycle in these. That is, that female matures, she'll lay a few eggs as she reaches sexual maturity, but once she's fully mature, ripe, she actually dies and becomes the chitin shell encapsulating 200 to 500 eggs within her. So rather than laying the eggs, she has become this heavy, hard chitin component to protect all of these eggs. All we have to do is put one microscopic pinhole through that cyst, and we just rendered all of those eggs useless. All we have to do is change the humidity in that environment, and that entire generation inside her is now rendered useless. When we got to Idaho and started playing with this, we realized that this wasn't a one-treat deal and we're done. We needed repeated applications to do this. I figured we've got this mapped out now. We'll run about a 25, 30-day treatment window every month. We're applying the Nemasan. I'm sure if you guys contend with nematodes at all, run into the same thing. Some of these species that we have, they lay eggs in a cluster. So the female will lay individual eggs, but she lays them in a pile, in a cluster. So the first time that the chitinase comes in contact with these, we tear apart the outer layer of eggs. If we don't keep going though, all of those eggs inside are still viable life forms. And so this is why we found we go with like say a 25, 30-day treatment schedule now, and we've had fantastic results with this. I'll show you some stuff here in a little bit, but other nematodes will lay individual eggs in a line. If that's the case, we can get those really quickly. Yes?
But even with the multiple applications, we're still 20% of the cost of the other.
Oh yeah, we're a fraction of what the cost is for traditional treatment methods. And anybody has any questions as we go along? I realize that this is kind of weird, mind-blowing stuff. Don't feel like you got to be polite and wait till they interject anytime. Throw your hands up. Let me have it. As a plant elicitor, this again, as Eric went through this morning, we don't understand a lot of this stuff, what's going on yet in the plant, but they've kind of identified the cellular and molecular changes. They can be summarized in membrane depolarization, oxidative burst, influx and exit of ions such as calcium, activation of MAP kinases, chromatin and DNA alteration, increase in PR gene mRNA, PR protein synthesis, phytoalexin accumulation, lignification, and callus deposition. The lignification and callus is kind of a big thing. Your plant, you'll read, if you start digging into this, you'll read stuff where it'll talk that your plant will have a fungal receptor built into the leaf surface, or it'll have an insect receptor, so that when those things land, it knows that something has landed there. When you boil this down and get into this, all that is a chitin receptor. Because if this is a pathogenic fungi, it has a chitin component. If this is an insect of pretty much any way, shape, form, size, it has a chitin component to it. So when we apply the Chitosan, they're close enough related, we are triggering that chitin receptor on the leaf surface. The plant thinks that this might be, it doesn't know. It doesn't know what it is, it just knows we've got to go to work, whether this is a fungal pathogen, an insect. One of the responses that they've been able to measure with this is the increase in lignin production. And I realize if anybody's involved in the animal industry, cattle in particular, they're trying to make lignin a 4-letter word these days. We actually have alfalfa growers though in Nevada that will put us out a pint every application because their test numbers and quality are so much better. And we've been able to identify what we're doing there is that petiole connection between the leaf and the stem. we will strengthen that to the point that they don't get the leaf shatter while they're raking and baling now. We get far better leaf retention. Guys with flood irrigation scenarios, it's deadly obvious where you have done the test plot and the grower standard. After the alfalfa crop is off and you turn the water back on, you will visually see the difference in the pile of hay leaves at the end of the field. So this lignification, this is one of the things when I talked earlier, we could go cut the stubble off on our test plots on our check and visually see that difference. That's what's going on. Because if this is an insect attack, your plant wants to stay standing to make it to reproduction. So this is one of the things that's going to trigger is lignin production to stay stiffer, bigger, stronger. Again, if the nutrition is not in place for this, this isn't going to happen. So this is part of why I say you have a problem show up, you put on an application, we'll light the plant up in 7 days, 10 days, it's going to tip over and sick again, and now I'm in trouble. This time and again, this is any time we've run into a situation like that, petioles, plant sap, there's something you've got to have, especially the micronutrients in place to drive all these functions. We have found with in cereal grains, ethafon applications are a big deal in my neighborhood, Cerone, to shorten things down. We apply this on at flag leaf. You've got to get it on the flag leaf, but before the awns come out, you've got about a 3-day window to make this happen. There is a side effect that we were always aware of with the ethylene, but we never knew. We just knew in our handline fields was where it was we've known for years that before we fly Cerone, we have got to be wet. We have got to have finished up an irrigation. On our hand lines, that isn't always possible. And we've noticed that anywhere that the hand lines had already watered, when we made the application, we would come through with no ill side effects. If we were in front of the hand lines when we made that application, we would see what looked like drought or frost. The barley would kind of turn white and just peak it. And for several days, this would be deadly obvious. A lot of times this will show up on the yield monitor even. We've stressed that plant to where the ethafon, being a hormone, what we've started now, if we're on a wheat crop, we don't worry about that anymore if we're putting the chitosan down. The wheat is usually enough stiffer straw that it stands with the chitosan. That seems to push it over the edge. We don't worry much about wheat lodging on our place anymore. The barley, some of these varieties will get tall enough, it helps, but it's not a complete replacement. But we have found that when we incorporate the O2, the chitosan, in with the ethylene, it completely counteracts this. What happens is the ethylene causes the plant to lose function or control of its stomatal function. So ethylene being a plant hormone, one of the side effects of it will hold that stomata wide open for 24 to 72 hours. So anywhere that we were dry, hadn't watered yet, we just caused a drought. We will affect stomatal conductance. I got a slide here, we'll get into this a little bit more, explain how that kind of works. But some of the places where we found that this has a very powerful fit on our farm. Nutrient mineral chelator. This is both good and bad. There's a few caveats to working with chitosan that you all need to be aware of. This is an extremely active cationic molecule, very positively charged, always looking for a negative